Welcome to the Unit 6 notes video on meiosis. The good news is this is one of the shortest units of the year. It used to be part of the unit that combined with mitosis. It was kind of confusing, a lot of the same terms and similar stuff, so we decided to split it up. It also comes with more good news that signifies that we are pretty much on the home stretch for the semester. And other good news is we're going to talk about sex. No, not really. We're going to talk about sexual reproduction, though. There is a difference. If you're not sure what that is, look it up. Don't ask me in class. It'll get kind of awkward, especially in front of everyone. So you can see there the first objective is the purpose of meiosis, its relationship to sexual reproduction, which is going to be pretty important for this unit, but also we're going to talk about it in a little more detail when we get to genetics and especially in evolution. We're going to talk about the eukaryotic chromosome. Once again, you've already seen some of this stuff in mitosis, but we're going to talk about it more in relation to now meiosis. And we're going to talk about genes and alleles, some kind of new vocab. We're talking about homologous chromosomes. Again, more just new vocab, but some stuff that should pretty much make sense since we've already started to cover some of this stuff in previous units. Number 4, summarizing the order of events in the phases of meiosis. Guys, this is going to be the most important objective for the test, and it's going to consist of about half of your notes that you have. This is going to be similar to the last unit where mitosis was a major focus. Again, this is only meiosis, so this is going to be the major focus of this unit. So if you have your highlighter out that you should be having out during this, you should highlight number 4 or circle it or something. That's going to be pretty important. We're also going to talk about the importance of an event called crossing over. We're going to talk about how meiosis relates to fertilization. We're going to compare and contrast some vocab terms, diploid and haploid. We're also going to talk about the differences and similarities between mitosis, which we've already covered, and meiosis, which we are covering in this unit. So let's get started. So objective 1, the purpose and importance of meiosis. Well, first of all, it's to produce sex cells, which obviously is important for life on Earth because of sexual reproduction, and we're going to get into a little bit of the details in this unit. Something that we should recognize for meiosis is that it has to take place pretty much perfectly. If there are mistakes that can happen, or that if mistakes do happen, genetic disorders can result in the developing offspring if the embryo does become an embryo. We'll talk about that a little bit later. The most important part of meiosis is that it creates variety in sexual reproduction. Again, we'll talk about a little more of this during evolution, but basically the result of meiosis is 4 genetically different daughter cells. In mitosis, we had 2 genetically identical daughter cells. That would be like a skin cell copying itself, bone cell copying itself, and so on. So we're going to talk about, and meiosis and mitosis are going to see a lot of similarities, but there's one big difference between them. So something that we're going to kind of get into a little more in genetics, but if you think of your own family or other people's family that have 2, 3, 4 siblings, brothers and sisters, well, unless they're identical twins, they probably look different. And the reason for that difference primarily is this unit, meiosis. So that's your answer for that fill in the blank that you're not going to give to anybody that didn't do their notes because they're slackers. So we're going to talk about the significance of sexual reproduction when we get to evolution again, but for now, just kind of an introduction to that. Next objective is about genes, alleles, and chromosomes. And again, when you see a double chromosome, that's one that has been copied. It's been replicated during S phase of interphase. And chromosomes, which we kind of started talking about, contain genes, and they're represented by letters typically in a chromosome. So these guys right here on the page are going to transform into what you see on your page. And so these are 2 homologous chromosomes. We'll get into some details of which. But guys, basically the genes that we talk about, hair color, eye color, size of certain bones representing your height and whatnot, are all just basically units inside the DNA that are things that we inherit from our parents that control our traits. So these things that you're looking at here are, like I said, are homologous chromosomes, and basically what that means is they share the same basic genetic information, such as if A were to represent something like a hair color, big A versus little a. We'll talk about genotype and phenotype a little bit. But basically, what you're looking at here are the genes, which we call the genotype. Okay, for instance, if this homologous chromosome on the left was from mom, and this homologous chromosome on the right was from dad, you can see how the genes differ slightly. In other words, maybe mom has dark hair and dad has light hair. You know, same letter A, but just lowercase and uppercase difference, showing the difference in the hair color. Same thing for B. We could say B is for eye color, dark eyes or brown eyes being capital B, and blue eyes being lowercase b. So you can see where the information is located on the chromosome, but slight variations, and those are the alleles that we talk about. The alternative forms of genes are called alleles, such as dark hair versus light hair, dark eyes versus light eyes, and some other things. Examples are there's millions of them. Skin tone is an example. We'll talk about some basic traits during genetics, such as being able to roll your tongue or not is thought to be a simple dominant or recessive example of genes that we have in our bodies, or alleles that we have in our bodies. Next objective is the big one, the order of events of meiosis. So knowing all the phases and what occurs during those phases. And since we have already talked about mitosis in enough detail that you should have a basic understanding, we're just going to add a few things to meiosis, and basically the rest is the same. One big difference here, meiosis has 8 total phases, whereas mitosis just had 4 PMAT. Meiosis has 2, PMAT and then PMAT again. So you're going to see 2 prophases, 2 metaphases, and so forth. The biggest difference between meiosis and mitosis that we'll see as far as pictures and events that occur happen during, for next fill in the blank here, during prophase. But it's not just prophase, it's prophase 1. And you'll see the difference here when we go through all the phases. But prophase 1, meaning the very first phase of meiosis overall, of all the 8 phases, prophase 1. You can also put prophase of meiosis 1 or prophase 1 of meiosis 1. There's kind of 2 meiosis C's, 1 and 2. A synapsis, we'll see a picture in a second, but a synapsis is basically the event that occurs when 2 homologous chromosomes, such as these guys, get really close to one another and sort of wrap up with each other during prophase 1. Okay, synapsis is where the homologous chromosomes get really, really close to one another. So they're touching. They're actually going to exchange DNA down here. We'll talk about in crossing over. The tetrad is just simply the object, the formation of homologous chromosomes being really close together. It's called the tetrad, and you can see why 1, 2, 3, 4, separate chromosomes that are all going to kind of interact with each other. Crossing over is the event that we're going to talk about, pretty important part of meiosis. And again, this is all during prophase 1 only. Crossing over is the actual exchange of DNA that occurs, okay, during a synapsis. Okay, when the chromosomes get really close together, they can and do very often exchange information, exchange genetic information. Okay, so here we have a picture of prophase 1, and you can see how there's a difference in the chromosomes. We're going to say these gray ones on your page, but you'll see them later on as pink, are going to be from mom, and the black ones we'll just say are from dad, just so you can see a distinction between the two, even if you don't color them, although I suggest you do on your notes. And so what we're talking about for crossing over is focused on this part right here, where these homologous chromosomes, again, they have the same genetic information, eye color, hair color, stuff like that, that they can exchange with each other. You can't exchange eye color for toenail size or something. Okay, this all has to be the same information that's getting exchanged. So let's just briefly kind of zoom into this crossing over event. So basically we're taking these 2 homologous chromosomes here. We're going to break them down like you saw earlier with these letters. Okay, and again, this crossing over is simply an event where homologous chromosomes exchange DNA. They exchange genes with each other. And that's where a lot of the variety comes into, is introduced for sexual reproduction, which is a humongous part of the evolution of life on our planet. So here we're going to say that the black homologous chromosome on your screen is going to be from dad. The pink one will be from mom. Can't get any more sexist than that, right? So here's a homologous chromosome from dad and homologous chromosome from mom. All right. Again, these are two homologous chromosomes. They have the same basic information. One might be for dark hair, one might be for light hair, but overall A represents hair color. Okay, so what happens during crossing over, which is basically this arrow right here? During crossing over, you see that they can exchange information. So now technically still dad's chromosome, has now picked up some of mom's, and since they've traded, that means mom has picked up some of dad's genetic information. So they switched it up. And you'll see when meiosis is over, completely over, these are the four, 1, 2, 3, 4 separate individual chromosomes that will result in 4 different cells. And so you can see right away you have differences. You know, no matter how you slice it, these are 4 different single chromosomes when they finally split from each other completely. So here are the phases of meiosis that we're not going to go through the details in this video, but you can see kind of like mitosis, they're going to line up in the middle. However, now they're lining up 2 by 2. During anaphase, they're going to separate from each other, just like in mitosis, but now since they are lining up 2 by 2, they're going to separate just how they lined up. And then during telophase, again, similar to mitosis, but now you can see that there's that genetic variation that exists now because of crossing over that took place during prophase 1. And then during meiosis 2, after this telophase splits, during meiosis 2, you're going to see these 2 cells. And they're each different, genetically different because of crossing over and because of some other stuff that you will hopefully have gotten in class or in the meiosis video. And again, during metaphase 2 now, this is so going backwards, prophase 2 and metaphase 2. Now you have 2 cells, metaphase 2, 2 cells. You can see they're slightly different genetically. Anaphase, they're going to separate, and this is just the same, this is very similar to mitosis. If you didn't see any crossing over taking place, this would look identical to mitosis. So metaphase 2 and now anaphase 2, again looking very similar. If it was black and white, it'd be identical to mitosis without this crossing over. Telophase 2, we have 2 cells that are going to separate into 4 cells. And again, the result of these 4 cells, you can see just by the colors, they're all different. And again, that's representing these guys here. These 4 individual chromosomes, which will eventually be located in 4 different cells after these guys split. Alright, so what this all has to do with fertilization and sexual reproduction too is that meiosis is absolutely necessary for fertilization because literally it's producing the gametes that are needed for sexual reproduction to take place, for fertilization to take place. You need 2 sex cells. So meiosis also performs what's called reduction division. Reduction, like reducing the number of chromosomes, reduction, and then put a hyphen, division, like multiplication division, because we're dividing the chromosomes in half. So reducing the number and then dividing them in half. So it's called a reduction division. And this will kind of help you remember the 2 parts of meiosis, 1 and 2. So this reduction division is important for gametes, super important for gametes, and embryos, which will result from those gametes. So fill in this word right here, embryos, E-M-B-R-Y-O-S. Getting the correct number of chromosomes. In other words, we have 46 chromosomes, 23 pairs. If the sex cells that created us had too many or too few, we would have 45 or 47 chromosomes. Incorrect numbers of chromosomes in a gamete or resulting embryo has basically 2 major consequences. Either the embryo does not exist, it just terminates, so the person is never a person, or it can lead to genetic disorders. And these other 2 slides we will cover in class.